Hello and welcome. This is our first video in the immunology section. Let's talk about structures involved in immune regulation. So, lymphoid structures are divided into primary and secondary lymphoid organs. Primary lymphoid organs are the site of creation of lymphocytes. These include the bone marrow and the thymus. On the other hand, secondary lymphoid organs are the site of proliferation of lymphocytes during an infection. These structures include the spleen, lymph nodes, pairs, patches, etc. Now let us see some of these structures and their importance. The first structures in this section are the lymph nodes. Here you can see a detailed labeled diagram of a lymph node. The lymph node is composed of the cortex, a paracortex, and a medulla. It is important to know where the lymphocytes are located in the lymph node. The outer cortex contains many follicles. These follicles are the site of B-cell proliferation. Primary follicles are dense and are usually dormant, while secondary follicles have a pale germinal center that are active. The paracortex, which is the region between the cortex and the medulla, contains the T lymphocytes. The paracortex also contains high endothelial venules, which are a gateway for the B and T lymphocytes to enter the bloodstream. The size of the paracortex increases in viral infection, while the size of paracortex decreases in DeGeorge syndrome. The medulla consists of medullary cords and medullary sinuses. Medullary cords contain the plasma cells, while the medullary sinuses contain macrophages. Okay, so the next structure we are going to talk about is the spleen. The spleen is located in the left upper quadrant, behind the 9th to the 11th rib, just anterior to the left kidney. It is divided into the white pulp and the red pulp. The red pulp is the hematologically active part that contains afferent arterioles that bring in the red blood cells, the macrophages that destroy the abnormal and old RBCs, and also the encapsulated bacteria. An important clinical correlation I would like to tell you about is when a person has asplenia, that is, the absence of a spleen, they are very prone to develop infections due to encapsulated organisms. The white pulp, on the other hand, is the immunologically active part of the spleen that contains periarteriolar lymphatic sheath, follicles, and a marginal zone. These store T cells, B cells, and macrophages respectively. Okay, so the next structure we are going to talk about is the thymus. The thymus is located in the anterosuperior mediastinum and is a derivative of the third pharyngeal pouch. It is the main site for differentiation and maturation of the T lymphocytes. So now let us see how a mature T cell is formed. As you can see in the following chart, a precursor T cell is made in the bone marrow. This T cell is not completely differentiated and has the ability to form both CD4+ and CD8+ T cells. Now, this T cell travels all the way to the thymus, where further maturation takes place. In the thymus, two types of processes take place: positive selection and negative selection. Positive selection occurs in the thymic cortex, while negative selection occurs in the thymic medulla. To know more about these processes, please watch our video on how positive and negative selection occurs in the thymus. Now, after positive and negative selection occurs, mature CD8+ T cells are released as cytotoxic T cells and mature CD4+ T cells are released as helper T cells. Further differentiation occurs in the lymph node by stimulations due to various cytokines. Depending on the cytokines, T helper cells can differentiate into Th1, Th2, Th17, T regulatory, and other such subtypes. I'll be covering the details of the functions of these subtypes in another video. Until then, thank you so much for watching. If you liked this video, share it with your friends and subscribe to our channel for more such videos, and don't forget to smash that like button.